வணக்கம். Welcome to NCERT Biology Reading. NCERT Class 11, Chapter 11, the second part video, 11.2 Plant Water Relations. Water is essential for all physiological activities of the plant and plays a very important role in all living organisms. It provides the medium in which most substances are dissolved. The protoplasm of the cell is nothing but water in which different molecules are dissolved and several particles suspended. A watermelon has over 92% water. Most herbaceous plants have only about 10 to 15% of its fresh weight as dry matter. Of course, distribution of water within a plant varies. Woody parts have relatively very little water, while soft parts mostly contain water. A seed may appear dry, but it still has water, otherwise, it would not be alive and respiring. Terrestrial plants take up huge amount of water daily, but most of it is lost to the air through the evaporation from the leaves, that is transpiration. A mature corn plant absorbs almost 3 liters of water in a dry, while mustard plant absorb water equal to its own weight in about 5 hours. Because of the high demand for water, it is not surprising that water is often the limiting factor for plant growth and productivity in both agricultural and natural environments. 11.2.1 Water Potential. To comprehend plant water relation, an understanding of certain standard terms is necessary. Water potential, that is psi W, is a concept fundamental to understanding water movement. Solute potential psi S and pressure potential psi P are the two main components that determine water potential. Water molecules possess kinetic energy in liquid and gaseous form. They are in random motion that is both rapid and constant. The greater the concentration of water in a system, the greater is the kinetic energy or water potential. Hence, it is obvious that pure water will have the greatest water potential. If two systems containing water are in contact, random movement of water molecules will result in net movement of water. Net movement of water molecules from the system with higher energy to the one with lower energy. Thus, water will move from the system containing water at higher water potential to the one having low water potential. This process of movement of substances drawn a gradient of free energy is called diffusion. Water potential is denoted by the Greek symbol psi or psi and is expressed in pressure units such as pascals. By convention, the water potential of pure water at standard temperatures, which is not under any pressure, is taken to be zero. If some solute is dissolved in pure water, the solution has fewer free water molecules and the concentration free energy of water decreases, reducing its water potential. Hence, all solutions have a lower water potential than pure water. The magnitude of this lowering due to dissolution of a solution is called solute potential or psi S. Psi S is always negative. The more the solute molecules, the lower more negatives is the psi S. For a solution at atmospheric pressure, water potential psi W equal to solute potential psi S. If a pressure greater than atmospheric pressure is applied to pure water or a solution, its water potential increases. If a pressure greater than atmospheric pressure is applied to a pure water or a solution, its water potential increases. It is equivalent to pumping water from one place to another. Can you think of any system in our body where pressure is built up? Pressure can build up in a plant system when water enters a plant cell due to diffusion causing a pressure built up against the cell wall. It makes the cell turgid. See section 11.2.2. In this increase the pressure potential is usually positive, though in plants negative potential or tension in the water column in the xylem plays a major role in water transport of a system. Pressure potential is denoted as psi P. Water potential of a cell is affected by both solute and pressure potential. The relationship between them is as follows. Psi W is equal to psi S plus psi P. 11.2.2 osmosis. The plant cell is surrounded by a cell membrane and a cell wall. The cell wall is freely permeable to water and substances in solution, hence is not a barrier to movement. In plants, the cell usually contains a large central vacuole whose contents, the vacuolar sap, contribute to the solute potential of the cell. In plant cells, the cell membrane and the membrane of the vacuole, the tonoplast together are important determinants of movements of molecules in or out of the cell. Osmosis is the term used to refer specifically to the diffusion of water across a differentially or selectively permeable membrane. Osmosis occurs spontaneously in response to a driving force. The net direction and the rate of osmosis depends on both the pressure gradient and concentration gradient. Water will move from its region of higher chemical potential or concentration to its region of lower chemical potential until equilibrium is reached. At equilibrium, the two chambers should have nearly the same water potential. You may have made a potato osmometer in your earlier classes in school. If the potato tuber is placed in water, the water enters the cavity in the potato tuber containing a countercurrent solution and containing a concentrated solution of sugar due to osmosis. Study figure 11.3, in which the two chambers A and B containing solutions are separated by a semi-permeable membrane. A solution of which chamber has lower water potential. B, solution of which chamber has the lower solute potential. C, in which direction will osmosis occur? Option D, which solution has a higher solute potential? E, at equilibrium, which chamber will have lower water potential? If one chamber has a psi of 2000 kilopascal and the other 1000 kilopascal, which is the chamber that has the higher psi? What will be the direction of the movement of water when two solutions with psi W is equal to 0. megapascal and psi W is equal to 0.1 megapascal are separated by a selectively permeable membrane? Let us discuss another experiment where a solution of sucrose in water taken in a funnel separated from pure water in a beaker by a selectively permeable membrane, figure 11.4. You can get this kind of a membrane in an egg. Remove the yolk and albumin through a small hole at one end of the egg and place the shell in dilute solution of hydrochloric acid for a few hours. The eggshell dissolves, leaving the membrane intact. Water will move into the funnel, resulting in rising in the level of the solution in the funnel. This will continue till the equilibrium is reached. In case sucrose does diffuse out through the membrane, will this equilibrium be ever reached? External pressure can be applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane. This pressure acquired to prevent water from diffusing is in fact the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent water from diffusing in. Numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. Osmotic pressure is the positive pressure applied while osmotic potential is negative. 11.2.3 Plasmolysis. The behavior of the plant cells or tissues with regard to the water movement depend on the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic, and if the external solution is more concentrated, it is hypertonic. Cells swollen in hypotonic solution and shrink in cells swell in hypotonic solution and shrink in hypertonic ones. Plasmolysis occurs when water moves out of the cell and the cell membrane of a plant cell shrinks away from its cell wall. This occurs when the cell or tissue is placed in a solution that is hypertonic, has more solids to the protoplasm. Water moves out. It is first lost from the cytoplasm and then from the vacuole. The water, when drawn out of the cell through the diffusion into the extracellular outside cell fluid, causes the protoplast to shrink away from the walls. The cell is said to be plasmolized. The movement of water occurred across the membrane, moving from one area of the high water potential, that is, the cell, to the area of lower water potential outside the cell, figure 11.5. What occupies the space between the cell wall and the shrunken protoplast in the plasmolized cell? When the cell or tissue is placed in an isotonic solution, there is no neat flow of water towards the inside or outside. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. When water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. The process of plasmolized is usually reversible. When the cells are placed in a hypotonic solution, higher water potential or dilute solution as compared to the cytoplasm, water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall. That is called turgor pressure. The pressure exerted by the protoplast due to entry of water against the rigid walls is called pressure potential psi p. Because of the rigidity of the cell wall, the cell does not rupture. This turgor pressure is ultimately responsible for enlargement and extension of growth of cells. What would be the psi p of a flaccid cell? Which organism other than plants possesses cell wall? What would be the psi p of a flaccid cell? Which organism other than plant possesses cell wall? 11.2.4 Imbibition. Imbibition is a special type of diffusion when water is absorbed by solid colloids, causing them to increase in volume. The classical example of imbibition or absorption of water by seeds and dry wood. The pressure that is produced by the swelling of wood had been used by prehistoric man to split rocks and boulders. If it were not for the pressure due to imbibition, seedling would not have been able to emerge out of the soil into the open. They probably would not have been able to establish. Imbibition is also diffusion. Since water movement is along a concentration gradient, the seeds and other such materials have almost no water, hence they are absorb water easily. Water potential gradient between the absorbent and the liquid imbibed is essential for the imbibition. Imbibed is essential for imbibition. In addition, for any substance to imbibe any liquid, affinity between the absorbent and the liquid is also a pure requisite. Thank you.